our webinar, so we will be introducing our fellows from the UST. So first one, we'll talk on cave microbiology research in the time of COVID, defying adversities in the midst of pandemic. So our speaker will be Mr. Nathaniel Alcantara, a graduate fellow. So he completed his Bachelor of Science degree in BS in biology, major in microbiology, and his Master's of Science in microbiology, minor in natural resource conservation at UPLB. He was also a visiting graduate student at North Carolina State University in USA, and currently he is a graduate fellow under the Career Incentive Program of the Science Education Institute and UST. He is currently working with the UPLB Museum of Natural History in their UST-funded research program on cave ecosystem. So I think Mr. Alcantara has prepared a video presentation.
Magandang umaga po ulit sa ating lahat. Ako nga po ulit si Nathan Alcantara. I am an MS Graduate Fellow under the Career Incentive Program of the Science Education Institute, Department of Science and Technology, and I am deployed in the UPLB Museum of Natural History. So I started my CAP Fellowship October last year. So halos isang taon na po. And for this morning, nais ko pong ibahagi yung naging experience ko po with this program. So first of all, Career Incentive Program is an initiative of the Department of Science and Technology through the Science Education Institute. This program specifically or primarily aims to provide training opportunities for their scholar graduates by deploying them in different research institutions in the country. So in my case, I am deployed in the UPLB Museum, and I'm particularly involved in their cave research program on Project 4, which is on phenomic, genomic, and metagenomic analysis of microbiome of botcat and guano from caves in Calabarzon. Actually, before I joined the museum, most of my research activities dealt with microorganisms associated with plants, such as banana, and more recently on pitcher plants. So when I learned about the project, I felt this is quite different from what I used to do. So I thought that this is going to be challenging and at the same time exciting for me, because handling plant samples for microbiological examination is very different from handling animal samples for the same purpose. For instance, for plants, if you need to collect roots, medyo madali lang. Hindi naman kasi umaalis yung halaman. Kung nasa siya nakatanim, nandun lang. If you need to cut them, wala naman masyadong issue. This is different for animal samples. Sometimes it can be very challenging to capture them, tulad ng paniki na lumilipad. And there's a lot of considerations when you need to collect samples from their internal organs, when you need to sacrifice them. So, yung project, it started last year. We are still in the midst of pandemic, and all of us are greatly affected. Hindi lang yung mga researches that involves microorganisms, but at the same time, other researches in other fields are also affected. We were placed in national lockdowns. We cannot do field activities. We cannot collect samples. And for us as microbiologists that need to collect samples from bats, this became more challenging because there were claims that coronavirus, which causes the COVID-19, actually originated from bats. And sa project namin, we'll be handling bats. At the same time, we'll be collecting microorganisms. Coronavirus is a virus which is a microorganism. So there were a lot of fear and people are very extra cautious. And to worsen the situation, during that time, we actually do not have a laboratory where we can do our work. And a laboratory or lab space is very critical for any microbiological experimentation because we need to practice aseptic technique. And so, kinailangan namin humiram ng facility sa iba't ibang units sa UPLB. And because our samples are coming from bats, we were denied several times. So it's very challenging for us. But eventually, we're able to find a lab and a solution to which we could tackle the problem. So we're fortunate enough that the Forest Biological Sciences Laboratory at the College of Forestry and Natural Resources allowed us to do certain portions of the experiments, as well as the Institute of Biological Sciences Microbiology Division. Eventually, they allowed us to do certain portions of the experiments under certain given conditions. So our strategy was to set up an on-site processing tent where we could do our initial work. So this is how it looks like. So basically, it's a big tent with two divisions. So there's an outer division where we place our materials, and there's an internal or an inner portions where we do our initial sample processing, where we maintain aseptic conditions, and we wore our PPEs, and we use sterile materials, and we maintain the septic condition in the area. So that became our strategy, and because of that, we were able to collect our samples from different sites in Calabar zone, such as in Cavinti. This is the area where we collected our first set of samples. Most of these photos actually from Cavinti, but eventually we were also able to collect samples from Quezon, Sungwan Cave in Quezon, and more recently in Labo, Batangas. And aside from that, we were able to form partnership with different institutions such as the Osaka University in Japan, as well as with Pascual Pharma Corporations. And we were also able to train students under an internship program. So these are two of the students I directly supervised during their internship. And fortunate enough, part of the results of their activities were accepted in the Asian Conference on Lactic Acid Bacteria, which is happening 10 days from now. The title is on the first Philippine report on lactic acid bacteria associated with Philippine endemic rat, Catus everata. So what's also interesting to note about it is that lactic acid bacteria is actually not part of the original proposal, when rats are actually also not part of our original source of samples. So this abstract or this study is actually a side study of a side study. And aside from that, we're also able to submit an abstract for an oral presentation in the International Science, Technology, and Engineering Conference, which will be held in December this year. And this abstract was also accepted. So we were able to train students, we were able to produce outputs, and we were able to form partnerships despite all the challenges that we faced in the first half of the project. And this is because of the efforts of our team members and their dedication to the project. So here we have on the left side, Juanita Tool, our University Research Associate, Director Marian, who serves as our project leader. That's me in the middle, and that's Professor Myla Anacleto from the Institute of Computer Science, and Ero Benitez, and Prof. Noel G. Sabino. We have other team members mainly from the Institute of Computer Science and from Institute of Biological Sciences. So despite all the challenges that we faced in the first half of the project, I think I can say that we were successful enough to be able to achieve our initial goals. So I think that's all. Thank you.